Hello, here we start with chapter 17. Okay, in the chapter 17 we are going to see the cytoskeleton, and whenever we talk about cytoskeleton, it's actually all about movement. So, movement is our starting point. Any organism, smaller or bigger, has to move outside because we are living in a dynamic world. Also, inside the cell, there has to be some certain movements. The cell is a complex soup of organic chemicals, all this organic material, and all this organic material has to be distributed, has to be reacted and compartmentalized. Of course, if you're talking about bacteria, this compartmentalization is not true. It's mostly about the aggregation. But if we're talking about the eukaryotic cell, we know that the surface over volume ratio for the eukaryotic cell is too small, so that in these large cells, diffusion is not enough. So maybe once upon a time, when all the cells were as small as prokaryotes, diffusion was a possibility. But when we come to these large cells, diffusion is not a solution anymore. So this is why the eukaryotic cell has to develop some compartmentalization and transportation systems. We have seen some of these transportation systems through the nuclear power complex, the endomembrane system, the mitochondrial transport, the peroxisomal transport. And of course, there are ways going in and coming out. And of course, there are more than one way. You can think that the bacteria, like the village, a bacterium can be a village, while a eukaryotic cell is Istanbul. So just like that, in a village, you can walk from one house to another house. Walking is a solution. In Istanbul, you cannot do that if you want to start from Büyükçekmece and end up in Pendik. Of course, walking is not a solution. You have to use one of the routes available, and there are different routes available. There's metro, metrobus, and in specific places, there's also the ferries going through them. Just like that, there are ways that you can use in a eukaryotic cell, and these ways in the eukaryotic cell, they're composed of cytoskeletal elements. So all these highways in a eukaryotic cell, they are made of cytoskeleton. Okay. Also, one more thing. We know that the prokaryotic cells, they are keeping their shape because in addition to their plasma membrane, they have the cell walls around them. The cell wall is thick. It's not made of protein, it's made of peptidoglycan, and it keeps the shape constant. But when we come to the eukaryotes, they are mostly lacking the cell wall. So many of the eukaryotes have no cell wall, but just a plasma membrane here. Because of this, they can change shape, and we know that changing shape is very useful because with this shape changing, they can eat other stuff, phagocytosis. And this was quite an advantage because they can feed on other smaller organisms, but also it has a price because it is not supported from outside anymore, it has to be supported from inside. So inside the eukaryotic cell, there must be elements which is keeping the consistency of the cell, which will allow the eukaryotic cell from breaking up, which will resist the stress, and which will allow this shape changes. And we know that these are cytoskeletal elements. So the cytoskeletal elements keep the eukaryotic cell strong, plus allow these small changes. So we know that the cytoskeleton is made of proteins, protein monomers. And these protein monomers can be reorganized. That's the whole idea, so that you can have this change shape. Okay. And if you check a eukaryotic cell, this is what it looks like. Here you see in the nucleus, around the nucleus, you see the microtubules all around. So microtubules start from the middle of the cell and they go outwards, and actin is usually just beneath the plasma membrane, and here the intermediate filament is not shown. Okay, there are three types of cytoskeletal elements in a eukaryotic cell: intermediate filaments, microtubules, and actin filaments. Okay, these are like different highways, just like in Istanbul you have metro, metrobus, and ferry, and sometimes other highways. In the eukaryotic cell, you also have these different kind of cytoskeletal elements. Their localization is different, their function is different because their chemical properties are different. Okay, their mechanics are different. They are going through different polymerization and depolymerization processes, and these tiny differences in their monomeric structure creates lots of big differences. Now, these intermediate filaments, we call them intermediate because they are thinner than microtubules, check the size, and thicker than actin filaments. So intermediate filament is the first thing. Now, check this nice lady. I guess she's very, very old. So her eyes and lips, everything is gone. And what is left is a little bit of skin and lots of hair. Now, all through these hundreds and thousands of years, she didn't lose the hair and the skin because these are mostly intermediate filament-based structures. So we can say that intermediate filaments are the most resistant cytoskeletal elements. So they can even resist to the pH differences, aging models, other reactive chemicals. So okay, this is one type of intermediate filament, but there are other types of intermediate filament in a cell, and I can say that intermediate filament is the strongest thing. It is the strongest because it can bend. It is not broken. Also, it resists to the mechanical stress. Think about a man. This man is standing up, and on the feet, there is like 80 kilogram of pressure, and the cells under the feet, they resist this 80 kilogram of pressure because it is full of intermediate filaments, and these intermediate filaments, they are resistant to mechanical stress. Okay, so that's what keep our tissues strong. Also, they are able to bend, they are flexible, and they don't break. Okay, this is also kind of very useful because you can stretch them. When you have the arm, you can stretch the tissue like this, and when you push it back, it comes back to the original position. It's because of some flexible molecules in the extracellular medium and in the cells. So these intermediate filaments, they are quite flexible. You can pull them, you can push them. They always go back to the original position. Now, in the cell, they originate from these small regions called desmosomes. Oops. Here, these are the desmosomes. And remember, there are some cell-to-cell connections. Here there are the tight junctions. Tight junctions are really weak. They're not strong enough to keep cells together, but they are tight enough to prevent passage of aqueous solution from one phase, which is now apical, to the other, which is now basal. So they have different purposes. Now, desmosomes, they are connected to intermediate filaments in the cell. So desmosomes are like nailing one cell to the other. Okay, this is why we have a continuous skin. This is why it is difficult to damage a skin. And if you check the intermediate filament staining in the cell, of course, this is a fluorescent microscopy image. You can see that from points they are connected and the intermediate filaments are continuous. This is why the cells make a continuous sheet. This is why they are anchored to each other. Also, this is why they can anchor to a surface. Here there's an electron microscopy. You can see the intermediate filament bundles and the desmosomes. Desmosome is a very nice structure. There are some transmembrane proteins here. And these transmembrane proteins, they are attaching head to head in the intercellular environment, in the intracellular environment, you can see the intermediate filaments attached. Okay, let's check the monomer. Like all cytoskeletal elements in eukaryotes, the intermediate filaments, they consist of protein monomers, and these protein monomers are filamentous, which means they are long and thin. There are many proteins in our body. They are mostly globular. These are not globular. These are filamentous. There are two domains here. The alpha helical region here to here is more or less the same between different kind of intermediate filaments, but there are some terminal regions, and these terminal regions, N and C, they have unstructured domains. These unstructured domains, they can have any shape. They can coil. They are not alpha helix or beta sheet. They can get very strange shapes. And this is the main difference between different kind of intermediate filaments. Now, when two monomers come together, they make a dimer. And in this dimer, the rod domains are connected to each other. And the unstructured domains are here again at the tip. Now, at this point, please notice something. The N terminus is here, the C terminus is here. So this is polar. It has two different directions. When you check the dimer, there is still an N terminus and there is still a C terminus. But when you check a tetramer, so here are the terminal domains. The alpha helix structures are here, and please notice that the two dimers are in opposite direction. So N terminus is here for this one and here for this one. So when you check the tetramer, you're going to see that it is N terminus here and N terminus here too. And the C terminus is hidden inside. So while this is like a vector N to C, this is not a vector anymore. This is an identical element. And these two ends are the similar, and this is the reason that intermediate filament is not polar. And when I say polar here, please don't think that it is like chemical structure. It is like hydrophobicity or hydrophilicity. No, it has nothing to do with that. When I say polar, I totally mean direction. N terminus here and N terminus here. And because it is not polar, it is not used for transportation. Okay, that's the main reason. Because for the transportation, if a molecule is walking on another molecule, it has to know which direction it's going. Okay. Of course, tetramer is not the final structure. Tetramers come together, 8 of them come together. Again, the N terminus outside and the unstructured region, they are penetrated here. And the tetramers are attached to each other, making a filament. And these filaments, they join each other to make fibrils. At the end, you see these very large structures, very resistant structure. So what keeps the dimer together is non-covalent bond between the monomers. What keeps the tetramers together is again the non-covalent bonds. And what keeps the tetramers, 8 tetramers together is again the non-covalent bond in this unstructured region. So although we know that non-covalent bonds themselves, they are very weak, altogether you have a very big number of them. And all these non-covalent bonds in a fiber and in a fibril too, they keep the intermediate filament in a very resistant way. And of course, this is a quite flexible structure. This is just an alpha helix. And we know that the alpha helix can be bent, as also you can see here, they bend a lot. This is why they have this elastic shape. Okay. Now, in the human body, there are, not in the human body, in the animal body, there are many different kinds of intermediate filaments, but overall, we can divide them into two, nuclear and cytoplasmic. Okay, let's start with the cytoplasmic ones. Oh, sorry. I'm sorry. Yeah, the cytoplasmic ones, the keratin is very famous. Let's see the keratin. Our hair and fingernails, they are made of keratin. The horns are made of keratin. The claws are made of keratin. And think about the claws of a cat and the claws of an eagle. They are both made of keratin and they have different features. So what makes hair, horn, and claws so different from each other, if they are all keratin, is that the alpha helical region is more or less the same, but the unstructured regions I'm sorry, are different. So how the unstructured regions are attached to each other here determines the strength of the keratin. So if the unstructured regions here are attached to each other much stronger, at the end it is going to be rigid like a horn. If the unstructured regions, they are kind of weakly attached to each other, it is going to be a weaker, easily bent structure like a hair. Okay. Also, the reason that why some people have curly hairs and some people have a straight hair is totally about the structure of keratin. Now check the keratin monomers and you're going to understand what kind of hair it is. Okay. The second class is vimentin. It is in connective tissue, muscle, and glial cells. We know that the glial cells can be very big and they have to give the structural support to the neurons in addition to every kind of nursing. So they have to keep their shape constant. They have the vimentin as intermediate filaments, what keeps their shape. And finally, there are neurofilaments. So if we check the axon, for example, what keeps the axon really strong is that they have some neurofilaments, which is actually kind of intermediate filament. So all around here, you're going to see them. So they give the consistency and they give the strength of the axon. So before you eat meat, you have to take out the nerves. Otherwise, it's going to be strange eating the meat that way. It's like long ropes, difficult to chew, difficult to digest. It's because of these neurofilaments. But yet, they are not used as the means of transport. The means of transport here is the microtubules, these green things. Okay. They are the highways, and these are the cement. Okay, of course, the any kind of problem with the keratin would cause weakness in the tissue. In this case, for example, the one of the keratin filaments is disturbed, and these people have this kind of severe wounds in their feet. Of course, not all of the keratins are disturbed in these people. Otherwise, they wouldn't be able to stand up, they wouldn't be able to develop or grow, survive. Only one kind of keratin is disturbed in them, and see what is happening. The tissue is torn because keratin is giving the strength here. This disease is called epidermolysis bullosa complex. And this is a kind of famous disease. The other one, progeria. Okay. These children, they get old extremely fast. Their aging is like tenfold more than the other people because the nuclear filament, they have the last class of intermediate filaments. They are mutant. The nuclear lamina here, it supports the nucleus from inside and it pushes the nucleus. This is why nucleus is always round. I mean, we never see nucleus like long and thin or damaged or something. Nucleus is always a perfectly round structure because of these nuclear lamina. Okay? And these nuclear lamina not only give shape, but also they are the attachment point to chromatin, which are the chromosomes. And if the nuclear lamina is not localized properly, the chromatin will not be able to attach. This is going to cause the DNA damage plus the transcriptional problems, because the weakly expressed genes, they have to be close to the nuclear lamina. The highly expressed genes, they have to be close to the center. So nuclear lamina, in addition to its structural properties, it has some functional aspect also. And without this, because they are having too many mutations and too many transcriptional problems, these children age extremely fast and they don't survive long. They die like 12 years old, unfortunately. And this is what the nuclear lamina looks like in the progeria disease. Unfortunately, it is detached really fast. Yeah. And finally, we know that there are different kind of cytoskeletal elements in cells. Here, for example, you see intermediate filaments and microtubules, and these two actually has to communicate. Now, in a healthy cell, the cytoskeletal elements, they have to be connected to each other for a stronger cell, because they have to support the plasma membrane from bottom. If they don't, plasma membrane will be simply ruptured, because plasma membrane is only as strong as olive oil. And you know that olive oil is not strong. It can easily detach, it can easily disperse. So here, for example, in red you see microtubule, in blue you see intermediate filaments. There are plectin molecules connecting them to each other, and this cross connection is very important. People have produced mouse mutant for plectin, and those animals didn't survive. I mean, they even died as embryos. They even couldn't finish their embryonic development. This shows the importance of not only cytoskeletal elements, but also their cross connection.